Okay, so now I'd just like to introduce our next speaker of the session, Dr. Yanchun Peng. Dr. Peng is a senior postdoc researcher working at the Human Immunology Unit at the Weatherall Institute of Molecular Medicine here at the University of Oxford. After completing her undergraduate studies and then teaching in China, she moved to the UK, first working as a research assistant and then doing her DPhil and remaining as a postdoc in the research group led by Professor Tao Dong since 2005. She's acquired extensive experience and skills in human T-cell immunology, with a major focus of her research being on evaluating antiviral efficacy of virus-specific T-cells and the interaction of host virus in HIV infection, HIV-HCV co-infection, and also influenza infection. Her recent interest is looking at viral oncoproteins and tumor-specific proteins, T-cells responses in virus-associated cancers, and she's used a number of cutting-edge technologies like multicolor flow, mass cytometry, single-cell RNASeq, with expertise particularly in isolating, expanding, and characterizing these virus and tumor-specific T-cells in vitro. With all of these different techniques, she really had to swiftly switch her work from cancer to COVID-19 as the pandemic started last year, and exploring the role of SARS-CoV-2 specific T-cells in COVID-19 infection. The title of her talk is The Role of Antigen-Specific T-cell Responses in SARS-CoV-2 Infection, so obviously a very common theme. And I'd like to hand over to Dr. Peng for her presentation.
Thanks for Jeremy. It's a great pleasure to share our COVID T-cell research work with everyone in this event. Today, I'm going to present our recent work, the role of antigen-specific T-cell responses in COVID-19 infection. So before presenting the data, I would like to introduce our COVID T-cell team, Dan Lim, Xuan, Tracy Guihai, and myself, led by Professor Tao Dong. We started working from the beginning of the pandemic and joined by postdoc Daniel and bioinformatician Lin, and also other lab members, Jilly, Dale, Magat, Wenbo, and Chen later on. So we know T-cells play an important role in controlling human virus infection. So T-cell can kill virus-infected cells through TCR-mediated signaling pathways. And the T-cell quality can be affected by multiple factors, such as antigenic variation, host genetics, and also microenvironments. The major determinants of an effective T-cell response are a viral antigen delivery. So what kind of viral antigen will be delivered to the T cells, and also the T cell function and the ability of T cells to kill. So the T cell function is mainly affected by the interaction of T cell receptor and peptide MHC class 1. The co-stimulatory and inhibitory molecules expressed on T cells also affect the T cell function to some extent. It is also well accepted that dysfunction of T cells can contribute to viral persistence. So how about COVID-19 infection? As we know, for COVID-19, the role of adaptive T cell immune responses in disease pathogenesis and long-term protective immunity is currently still poorly defined. But this information is essential to inform potential therapeutic interventions and to enhance vaccine design. So current vaccines aim to induce antibody responses to the spike protein, but it's not yet known whether this will be sufficient to induce long-term protective immunity to SARS-CoV-2. So in the situation of emerging virus variants, which already compromise the efficacy of current vaccines, study the natural immunity to the virus, especially the role of SARS-CoV-2 specific T cells, therefore is critical to fill the current knowledge gaps for improved vaccine design. So this just a typical kinetics example of kinetic T cell response during virus infection. Since COVID-19 is a new virus. We know very little about its nature and immunity. So we started very basic questions. Which part of the virus T-cell targeting? Then what is the immune memory like in patient recover from COVID-19? Is there any association with disease outcome? As we are expecting COVID-19 will be around for a long period, and the emergence of viral variants, it's also very interesting to know the kinetic change of memory response over time, whether they can protect us from secondary infection or against new emerging viral variants. So in order to answer this question, we characterize SARS-CoV-2 specific T-cell response in a very small patient cohort recovered from COVID-19 infection. So this data have been published by Nature Immunology last year. So in this paper, we found that broadly targeted T-cell response against SARS-CoV-2, and the total T-cell response is stronger and broader in severe cases. Our data also suggests a protective role of SARS-CoV-2 specific CD8 T cells. What we found is great proportion of CD8 T cells response to spike and MMP in mild cases, and a higher proportion of the multi-cytokine producing M and NP specific T cells is associated with mild disease severity. So then we further mapped this CD8 T cell response and identified 6 dominant CD8 epitopes, 5 from NP, 1 target ORF3, as you can see from this table, and the FACS plot stemming from the pentamers we get from ProImmune. So then we focused on this HAB7 restricted NP response, SPR response, and to explore the role of antigen-specific T cells in COVID-19 infection. The reason we did this is we found this B7 epitopes appears to be the most dominant epitope. There's no variance found within this epitope from over 300,000 sequences in the global bank. They also have unique features which revealed by Loin and Nierenberg recently. They published their findings in Immunity. They found that shared TCR receptors specific to this epitope were observed in SARS-CoV-2 unexposed and infected individuals. But however, those cells are naive cells, and they are unlikely to have resulting from previous seasonal coronavirus infection. So in our proof of concept study, we aim to investigate the correlation of this B7 restricted NP epitope with disease severity, and then explore their TCR diversity and also the potential mechanism underlying this protection. So unsurprisingly, in our cohort, 80% of B7 patients showed response to this dominant epitope. Among them, 6% were recovered from mild disease. Also, these people showed a significantly higher response to this epitope, and also the response to this single epitope accounting for average of 6% of total response to NP protein in mild cases when compared to the patients who had severe symptoms during early infection, showing like only 20% on average. So then we studied the this T-cell response in depth by our single-cell platforms, mapping their T-cell receptor repertoire and evaluating their functions over time. This workflow slide just showed how we did it. Briefly, we stayed PBMCs with MHC class 1 pentamer and sought the antigen-specific T cells at single-cell level. So part of cells went into cell lysis buffer for RNASeq. Part of cells were sorted into cell culture medium and expanded in vitro as T-cell clones or bulk lines. For RNASeq, the data analysis will reveal the TCR receptor usage and the gene expression profile of each single cell. Then in vitro expanded T-cell clones will be functionally evaluated, and TCR will also be sequenced too. So the in vitro and our in vitro and ex vivo data will be linked through the T-cell receptor. We'll only focus on those shared T-cell receptor between the clones and the ex vivo single cells. Finally, we'll just like assess the antiviral efficacy of those T cells by our in vitro viral infection assays to see whether there's any specific T cell subset could contribute to disease protection. So far we have done SmartSeq 2 single cell analysis with 4 patients, 2 recover from mild disease and 2 from severe infection. So these sucker plots just showed paired TCR alpha chain and beta chain usage, which showed like very broad TCR usage by both mild and severe patients, whereas a dominant famous influenza HA2 restricted M1 epitope have much narrower TCR usage. These just examples of from 2 healthy controls. So then we let's compare the gene expression profile of this B7 NP specific T cells between mild and severe. As you can see here, T cells isolated from patients recovered from severe infection express significant higher cytotoxic molecules, including granzyme A, B, K, granulizing, and also NKG7. Meanwhile, those cells also have accumulated inhibitory receptor expression, such as TIGIT, CTLA-4, and TIM3. And they also are more activated and well differentiated when compared to T cells from patients only suffer from mild symptoms. So as I mentioned, Lung and Lindberg observed those shared TCRs in SARS-CoV-2 unexposed and infected individuals. So is this the case for our study? If yes, is there any specific features for this group of T cells? So then we calculate the similarity scores of the CDR3 beta sequences from our COVID-19 patients to those healthy pre-pandemic individuals, which including all the published sequences from the 2 papers I mentioned previously, and also the data set from combat study in Oxford, showing this plot. So y-axis it presents the similarity score. So a score of 1 corresponds to complete similarity, means those T cell receptor is exact same sequences. And 0 just represent complete dissimilarity. Apparently, CDR3 sequences from mild patients are more similarity to those naive precursors found in pre-pandemic individuals. Then we group these T cells into high similarity group and low similarity groups, and look at the gene expression profile. As you can see, the cells with CDR3 beta sequences that are more similar to pre-pandemic individuals, they show high expression of inhibitory receptor molecules with upregulation of TIGIT, CTLA4, BTLA, and the TIM3. So as I mentioned, like when we did single cell sorting for SmartSeq 2, we also sorted some cells for in vitro T cell cloning and expansion. So we generated a bunch of T cell clones from these 4 patients we studied, and sequenced the T cell receptor of each clone and measured the EC50 by interferon gamma ELISpot as shown in this plot. Then we grouped the T cell clones and also the single cells from our SmartSeq 2 according to their TRBV gene usage and the CDR3 beta sequences into 18 groups as shown in this table. So each group with a unique TRBV and TRBG usage, and also CDR3 beta usage. So use the EC50 for the clones in each TCR group. We divide them, all the cells, including the clones and ex vivo single cells from SmartSeq 2 into 2 high functional avidity, showing cyan, and low functional avidity, the TCRs with low functional avidity showing red. And then we look at those TCRs, as I mentioned, like shared between COVID-19 patients and healthy pre-pandemic individuals. I want to look at, are they high avidity T cells or low avidity T cells? So our analysis showed that TCRs with high similarity scores to the ones observed in pre-pandemic individuals seem to have a lower functional avidity fed into this group. And we also evaluate the function of these T cell clones with naturally processed antigen using vaccinia virus and real virus infection. So firstly, we, by collaborating with Jeff Smith's group in Cambridge, we tested our T cell clones with the vaccinia virus expressing SARS-CoV-2 proteins. So we co-cultured T cells with autologous B cells infected with the vaccinia virus expressing SARS-CoV-2 protein, and in parallel with peptide-loaded target cells. Here are just examples of FACS plot from ICS showing the CD107A expression and cytokine production from those T cells responding to peptide and vaccinia virus infected targets. Although the response is much weaker with vaccinia virus infected targets when compared to the peptide, but the strength of the response to this naturally processed antigen is correlated with their functional avidity. When you look at these plots show the correlation between the CD107 expression and the EC50. So with the great help from facility in the WIM and different collaborators within the university, we managed to set up the virus infection system to assess the antiviral efficacy of SARS-CoV-2 specific T cells in vitro. So we transduced autologous EBV transformed B cell lines with lentivirus expressing S2 gene, then infected the with SARS-CoV-2 viruses, including Victoria strain and also other variants like B1.17 and others. And we co-cultured our T cells with those virus-infected target cells and assess the T cell activation by ICS. This FACS plot just showed the HAB7 NP specific T cells. They can recognize virus-infected target cells in vitro. And we also set up another evaluation system to look at whether those T cells can suppress the virus replication or not. So this just showed our preliminary data from the experiments, which showed our T cell clones we isolate from recovered COVID-19 patients can suppress virus replication, and their antiviral efficacy is correlated with their functional avidity, as shown this plot, the elective correlation between the EC50 and the percentage of virus suppression. So the last question is, how long will this protective T cell remain? So we collected PBMCs from patients at 6 months after COVID infection and pentamer sorted those B7 positive cells and did the T cell sequencing. This just show an example of T cell repertoire changes from months of patients we followed. As you can see, the T cell repertoire narrowed, but consisting both high and low avidity clones 6 months after infection, and separately, they are capable of controlling virus replication in our assessment. So to wrap up, data from this proof of concept study showed the memory B7 NP specific T cell response is dominant in COVID recovered patients and is associated with mild COVID illness. So our single cell analysis of pentamer sorted cells revealed highly diverse T cell receptor usage by these T cells, and also strong cytotoxicity and high inhibitory receptor expression is associated with disease severity. This may indicate a stronger antigen stimulation in severe cases during the early infection. So we also observed similar situations. The TCR sequences from mild cases showed higher similarity to those naive T cell precursors found in pre-pandemic individuals by other researchers. This suggests these cells might have the advantage of expanding earlier and faster in the early infection, as the data presented by Nina showed early T cell response is correlated with shorter symptoms and mild disease outcome. And our data also showed that TCRs with this high similarity to pre-pandemic sequences have higher inhibitory receptor expression. This may indicate the longer exposure to antigen during these early responses. Our in vitro analysis also showed that HAB7 NP specific T cell clones showed broad range of functional avidity. Interestingly, TCRs with higher similarity scores to those precursors found in pre-pandemic individuals, they seem to have a lower function avidity. So by using our in vitro vaccinia virus and rare virus infection system, we evaluate the response of B7 NP specific T cells to natural process antigen. We found out that strength of response is correlated with their functional avidity. The T cells with high avidity can suppress virus replication efficiently in our system. We also observed like the narrowed T cell repertoire, but consisting high and low avidity clones in individuals 6 months after infection. This suggests the proportional but not avidity dependent T cell memory contraction. So there's still lots of questions unanswered. Now, with rapid emerging of new virus variants, which has escaped antibody response elicited by natural infection and vaccination. So in that understanding what the correlates of protective T cells following SARS-CoV-2 infection or vaccination is needed for better vaccine or therapy in the future. So final slides for today's talk. The work I presented today is a great team effort involving a lot of people in Oxford and beyond. Many names may not list here. First, I would like to thank Tao for her great guidance and huge support and the hard work from our great T cell team. Xuan, Guihai, Zixi, and Danning for T cell work, Jili for helping with vaccinia virus work, Daniel for helping with ACE2 transduction, and Nim for all the single cell data analysis with the help of combat repertoire team in Oxford. Huge thanks go to Graham and all the facilities from HIU and within the WIM for the scientific and management support. Moreover, I would like to thank the Julian and Alex for their very valuable cohort. And many thanks to all our collaborators listed here, such as ProImmune for their pentamers, and Jeff Smith for Jeff's group for vaccinia viruses. Last but not least, great thanks to all the patients and their family, and also the funding bodies which made this study happen. Thanks for your attention. I'm happy to take questions.
Thank you very much indeed, Peng, much appreciated. We don't have much time for questions because I want to make sure that we have got some good discussion for the panel. There is one question from Yu Chol who's entered in relation to that B7 nuclear protein immunodominant epitope, the 106. How similar is that in regards to common cold coronaviruses in regards to homology, amino acid homology?
I think the epitope itself, they have like some like not conserved, but there are some several amino acid difference. Yeah.
Yeah.
Do you know how many difference among nine amino acid?
I can't remember, but from like Lewin's paper, they suggest that epitope from seasonal coronavirus may haven't been processed and presented. Yeah.
Great. Okay, so we're going to thank you very much indeed, Peng. That's fantastic.